How can a doctor solve the really complex mystery of anemia, sometimes almost instantly? Well, the answer isn't some high-tech scanner. Nope. It's found in a single drop of blood, and it's examined using one of the most powerful and, honestly, elegant diagnostic tools in all of medicine.
So that begs the question, right? What secrets can one little drop of blood actually reveal? Well, it turns out when you know what to look for, that single drop tells a vivid, detailed story about what's going on inside the body. And our investigation starts right now. So, the main piece of evidence in our whole investigation is something called the peripheral blood smear. You can think of it as the scene of the crime, a place that holds all the clues we're going to need to crack this case wide open.
So, what exactly is a peripheral blood smear? Well, it's pretty much what it sounds like. A thin layer of blood is smeared on a glass slide, it gets stained, and then it's examined under a microscope. But, for a pathologist, this isn't just a slide. It is a literal window into the entire story of a patient's anemia. Every good investigation has to start somewhere, with that first big clue. And for us, that clue comes from the complete blood count, or CBC. It's a value called the mean corpuscular volume, or MCV for short, and it just tells us the average size of the red blood cells. And this right here is what makes the MCV so incredibly powerful. It immediately simplifies everything. It takes the huge, complex world of anemia and splits it into just 3 possible paths. Are the cells too small? Are they normal-sized? Or are they too large? This one data point just narrows the possibilities down immensely. Okay, so with our 3 paths laid out, now we can start following the trails. We're going to dive into the blood smear and look for the very specific visual clues, the fingerprints, if you will, that each type of anemia leaves behind on the red blood cells. First up, let's go down the microcytic trail, where the cells are too small. Now, in iron deficiency anemia, you don't have enough iron to make hemoglobin. That's the protein that makes blood red. This makes the cells look small and pale, and sometimes they even get stretched out into these elongated pencil cells you can see on the right. But what if the cells are small for another reason, something like thalassemia? Well, the smear looks totally different. The key fingerprint here is the target cell. You see that bullseye shape? That happens because defective protein chains create an excess of cell membrane, which kind of bunches up in the middle. So here's the takeaway. Small, pale cells point to iron deficiency. Small cells with lots of targets, that points you towards thalassemia. All right, let's move on to our second path, normocytic anemia. This is where the cell size is normal. Now, this category is way broader, which means the visual clues you can find on the smear become absolutely critical to figuring out what on earth is going on. One of the biggest clues here is something called a spherocyte. It's a small, dense, spherical cell. Seeing these is a major sign of hemolysis, which just means red blood cells are being destroyed. They actually form when macrophages in the spleen take a little bite out of the cell's membrane, causing the rest of it to ball up into a sphere. Okay, now this is a dramatic and really urgent clue. These are called schistocytes, and they're literally fragmented red blood cells. They look like tiny shards of glass or little helmets. Seeing these means that red cells are being physically ripped apart as they travel through the blood vessels, usually by tiny clots. This points to very serious, often life-threatening conditions. Finally, let's head down the macrocytic trail, where the cells are too large. And on this path, shape is everything. If the big cells are distinctly oval, and you also see these neutrophils with way too many lobes, that is a classic textbook sign of a B12 or folate deficiency. But if the large cells are perfectly round, that points to completely different causes, like liver disease or alcohol use. It's a really subtle difference, but a vital one. So we've looked at size, we've looked at shape, but believe it or not, the investigation can go even deeper. Now we're going to start looking for hidden messages inside the red blood cells themselves. These are called inclusions. Think of these inclusions as secret notes left behind by specific disease processes. Each one tells a unique little part of the story, and sometimes finding just one of them can be the key that cracks the entire case. So for example, these things called Howell-Jolly bodies are basically leftover bits of the cell's nucleus. Normally, the spleen cleans these out. So if you see them floating around in the red cells, it is a huge clue that the patient either doesn't have a spleen or their spleen isn't working right. Or how about this one, basophilic stippling? These little fine blue dots you see scattered inside the cell are actually clumps of degraded RNA.
Seeing this specific pattern can immediately point an investigation towards very specific conditions, like lead poisoning or some types of thalassemia. So we've got all these different clues, right? Size, shape, color, and these secret internal messages. The final step is to put it all together. Let's take a look at how a pathologist synthesizes all this information to solve the mystery with incredible speed and confidence. It really all comes together in a very systematic 5-step approach. A pathologist starts with the MZV, then assesses color, then evaluates all the shapes and looks for any inclusions. The very last step is to check the reticulocyte count. That's just the number of new red blood cells being made to see if the bone marrow is responding properly. When you follow these steps, a clear diagnosis just emerges, almost instantly.
Okay, let's do a quick case file. We look at a smear and we see 2 big clues. There are fragmented red cells, schistocytes, and some of the cells have a bluish tint, which tells us they're really young. So what's the diagnosis? Microangiopathic hemolytic anemia. It makes perfect sense. The fragmented cells show they're being destroyed, and the young blue cells show the bone marrow is working overtime to fight back. The clues don't just give us the name, they give us the mechanism. Case closed. Let's try one more. Case file number 2. You look at this smear, and it is just full of really large, oval-shaped red blood cells. On top of that, the neutrophils have way too many segments in their nucleus. The diagnosis here is immediate: megaloblastic anemia. These 2 features together are the absolute classic signature of a B12 or folate deficiency. See? This systematic approach makes even these complex diagnoses seem clear and logical. Following this process leads to a fast, accurate diagnosis. But is that really the ultimate goal here? Is it just about slapping a name on the condition? No. The real goal is something way more profound. By mastering the art of reading a blood smear, you're doing so much more than just identifying a disease. You are peering into the very biological mechanism that's causing it. You aren't just solving a mystery, you're understanding its fundamental cause, all revealed in a single drop of blood.